Welcome to the next lecture in Bio 110. This is going to be covering the last set of subjects that we are going to be exploring in class about cell division and the cell cycle. In here you have a list of the objectives that we're going to be covering in class. I will like you to take a look at these objectives at home and be able to understand what are going to be the expectations about the following materials that we're going to be covering in class. Well, let's begin to talk about the cell cycle and having an idea of how the cell cycle is controlled. So as you know, all cells are derived from pre-existing cells. No cell is derived from spontaneous generation. So a cell comes out from the division of a mother cell into two daughter cells. Now this is a process, the cell cycle. So the cell cycle, it's orchestrated as a set of replication events where you're going to have duplication of cell material, and after that you're going then to have a division event where that material is going to be divided equally among two daughter cells. Now during the cell cycle, what we're going to have are material sources as the membrane, the organelles, the DNA are going to be replicated, and eventually they're going to be equally divided into the two daughter cells. Now the cell cycle is going to be controlled by a pair of kinases. So those are going to be called the CDK proteins, and they're going to be controlled by regulatory proteins called cyclins. What we're going to look is how the cyclins and the CDKs are going to be interacting with one another to control the cell cycle. As the cell cycle is progressing, you're going to have checkpoints, and those checkpoints are going to be stops in which different processes are going to be evaluated, and if they pass muster, they're going to allow the cell to continue with cell division, and eventually the cell will divide into two. Now, if there are problems during cell division, and those problems cannot be resolved during the checkpoints, the cell will be programmed to death, and that is involved during programmed cell death, which, as you may imagine, is very important for the organism, and dysregulations in programmed cell death have been, for example, shown to cause and induce cancer. Now, when we take a look at the stages of cell cycle, we're going to be looking that we can divide it into three major steps. We're going to be seeing one step that is going to be the cell growth and chromosome duplication step. So here we have a cell, and that cell has two chromosomes, one black chromosome and a red chromosome inside the nucleus. The material inside that cell, including the membrane and the organelles, are going to be duplicated, resulting in a much larger cell, shown here at the bottom. But also, you're going to have the duplication of the chromosomes. So we have here now two red chromosomes and two black chromosomes. Eventually, those chromosomes are going to be segregated, and each of the daughter cells will inherit an identical number of chromosomes. Once the chromosomes have been segregated into the daughter cells, you're going to have the event of cell division. So we're going to take a look more closely at how all these three different steps are related to the cell cycle. Now, when we look at the events that I just showed you up in the previous slide, what we have is that all those events can be divided into four different phases of the cell cycle. One of those phases is called interphase. An interphase is composed of three different phases. You have the G1 phase, or gap, or growth phase. That is going to be the initial step in which the cell is getting ready to divide. So you're going to have a lot of multiplication of components inside the cell. You're going to have the S phase, for synthesase phase, where you're going to have the DNA of the cell being replicated. After the DNA has been replicated, you're going to have another growth phase called G2 phase, where you're going to have organelle division. And at the end, when all the components of the cell are divided and the cell has grown, you're going to have N phase. And N phase is going to include nuclear division as well as cytokinesis. So you're going to have the process of mitosis as well as cytokinesis, which is the division of the two cells. Now, at every point in the cell cycle, the cell is always asking the question, has things gone wrong? Is the environment appropriate? And those are considered checkpoints. So for example, as the cell is going through the initial growth phase before it enters the DNA replication phase, it is asking, is the environment favorable to undergo S phase? And if it is, the cell will enter S phase and begin DNA replication. As DNA replication continues, and now the cell has finished DNA replication and begins to divide the organelles, the cell is going to ask, was all the DNA replicated? Was there any damage? And if it was that damage repaired? And if those steps are answered yes, the cell will then enter the M phase and enter mitosis. And during mitosis, we're going to see the alignment of the cells in the mitotic spindle, and that is going to be another checkpoint. Are all the chromosomes properly attached to the mitotic spindle and aligned in the middle of the cell? And if that is accomplished, then the cell will duplicate by pulling the chromosomes apart, and the cell cycle will begin anew. So the cell cycle checkpoints are going to be designed to make sure that the process of cell division happens flawlessly. So let's take a look at how this is controlled. So we're going to have the effect of cyclin-dependent kinases, and those cyclin-dependent kinases are going to be used by the cell to allow cell division. So those cyclin-dependent kinases work in pairs. You have a unique cyclin-dependent kinase that has to bind to a cyclin protein. That pair is unique, and when it's bound together, it initiates the activation of the complex. Now, you're going to have a cyclin that is going to pair with only one CDK, and that forms an inactive cyclin-CDK complex. Now, in the cyclin-dependent kinases in that cyclin-CDK complex are going to be phosphorylated in certain residues, and afterwards dephosphorylated in other amino acids, and once that process happens, that is going to induce activation. So once phosphorylated and dephosphorylated, that process is going to induce activation, and I'm going to show you that in a moment. At the end, the activated cyclin-CDK complex is going to work as a kinase, and that kinase is going to be able to phosphorylate other proteins that regulate cell cycle. Now, how are this cyclin-CDK complex discovered? So the work done that awarded the Nobel Prize in 2001 by the discovering of the cyclins was done on Xenopus. Xenopus is this little ugly frog here at the bottom. Actually, it's so ugly that it's cute. And its eggs, or its oocytes, are sufficiently big to allow us to understand a lot of biological process, so we can use them. So what the experiment did was to take cytoplasm from a cell that is undergoing mitosis and inject it into a Xenopus oocyte, shown here in gray. So now that cytoplasm from the cell undergoing mitosis, shown here in green, completely permeated the cytoplasm of the oocyte, and what you start to see is that the DNA in the nucleus was induced to form a spindle and a line. So the oocyte, even without activation, was driven to N phase. That was the indication that some component in the cytoplasm of a cell in M phase was required and significant to allow a cell that was quiescent, meaning non-divided, to initiate N phase. So for example, the control for that experiment was to take the cytoplasm of a cell that is in interphase, either because the cell is in G1 phase, S, or G2 phase. So taking the cytoplasm from a cell that is in interphase and inject it into a similar oocyte didn't allow the oocyte to enter into M phase. So this was the indication that some components within the cytoplasm of a cell in M phase, it's going to then and required to trigger N phase in the oocyte. And this is where the cyclins were discovered. Now, as I mentioned, you have cyclin-dependent kinases that, as the name implies, require to bind a cyclin, and this is shown here in this picture. So what we have are the cyclin-dependent kinases, and those are present in all cells in all the stages of the cell cycle. So they're always present in the cytoplasm of the cell. Now, The activity of the cyclin-dependent kinases is controlled by two things. Number one, they need to bind their appropriate cyclin, shown here in green. And once they have bound to their appropriate cyclin, the cyclin-dependent kinases needs to be phosphorylated in certain residues and dephosphorylated in others. So for example, M-CDK is a cyclin-dependent kinases that triggers M phase, and S-CDK, it is a cyclin-dependent kinase that triggers the S phase. Now the cyclins, as the name implies, are proteins that are regulatory to control cyclin-dependent kinase activity. And also, as the name implies, they cycle. They're not always present in the cell. So they're only produced during the appropriate phase of the cell cycle. And one example of those cyclins are going to be the M-cyclins and the S-cyclins. So you may imagine now that the M-CDK is going to bind to the M-cyclin and the S-CDK is going to bind to the S-cyclin. This table shows some of the major cyclins and cyclin-dependent kinases that are present in vertebrates. So in the left, in the first column, what you have is a cyclin-CDK complex. So you have, as you can see, one G1-CDK, that is important to the G1 state of the cell cycle, that is composed of cyclin D, and it could be composed of either CDK4 and CDK6. The G1S-CDK is another cyclin complex that is composed of cyclin E as well as CDK2. The S-CDK is composed of cyclin A and the CDK2, and the M-CDK, which is the one required for the M phase, is composed of cyclin B and CDK1. So as you see, different CDK partners will bind to different cyclins, generating different cyclin-CDK complexes. Now, this movie is going to illustrate some of the mechanism by which the cyclin-CDK cyclin-dependent kinases are going to be activated by the CDKs.
Cyclin-dependent kinases, or CDKs for short, are crucial regulatory proteins in the cell cycle. When activated, these kinases transfer phosphate groups from ATP to serine and threonine side chains on target proteins. When inactive, the active site of CDKs is sterically occluded by a loop, often referred to as the T-loop. As their name suggests, cyclin-dependent kinases are activated by cyclins. Cyclin binding to CDK pulls the T-loop away from the active site and exposes the bound ATP, allowing it access to target proteins. Thus, a CDK can phosphorylate target proteins only when it is in a cyclin-CDK complex. A third protein, called a CDK activating kinase, is required for full activation of CDK. This kinase adds a phosphate group to a crucial threonine in the T-loop, thereby enabling CDK to bind to and phosphorylate its target proteins. Target peptides bind to the active site of the cyclin-CDK complex so that the target serine or threonine side chains are precisely positioned with respect to the gamma phosphate of the bound ATP. CDK inhibitor proteins, or CKIs, help regulate the rise and fall of cyclin-CDK activity. Some inhibitors, like the one shown here, bind directly at the kinase active site and block kinase activity by interfering with ATP binding. Other inhibitors bind near the active site and interfere with substrate binding.
So that movie illustrated part of the requirements to activate a cyclin-dependent kinases. As the movie stated, you need to have the cyclin bind to the CDK to expose the active site of the CDK molecule. You also are going to require the molecule to be phosphorylated for activation. So now, this image over here, it's showing you how the different cyclins are activated during cell cycle. So for example, in here we have the M-CDK, which is the cycling that functions during mitosis. What we have here in pink, it's the level of the M-cyclin that, as you see, raises up during mitosis, comes down during interphase, and slowly goes up until it's peaked back in mitosis, and then drops down again at the beginning of interphase and starts to go up again. The cyclin-dependent kinase activity for the M-CDK is only shown during the state of mitosis and not during interphase. So that is indicating to you that when the M-cyclin is present, it will be able to bind to the CDK and activate M-CDK to have CDK activity. And it's only during the time where the cyclin is present that the CDK molecule is active. This illustration here, taken from the large Albert books, it's showing you how different cyclin-dependent kinases are activated during the cell cycle. So here in blue, you have the G1S cyclin. In red, you have the S-cyclin, and in green, you have the M-cyclin. So during the G1 state, you have a lot of G1S cyclin, and therefore that is allowing to form the G1S CDK during the G1 state. That goes immediately down when the cell progresses to S phase, shown here by the decline in the blue line. At the same token, during the G1 phase, you have the increased amount of S-cyclin that stays with you during the S phase of the cell cycle. The M-cyclin is not present during the G1 state nor the S phase. At the end of the S phase, it's beginning to climb up a little bit during the G2 state, and it peaks during M phase. And therefore, it is only able to bind and activate the M-CDK at the end of the G2 phase. So when you look at this table and this information, you can basically start to look at the activity of the different G1 cyclin-CDK complexes and see how the recycling of the cyclins allows for the activation of each of these kinase complexes. These complexes are activated by phosphorylation and dephosphorylation. So we're going to have two different phosphate sites in the cyclin CDK complex. One of those complexes is going to be phosphorylated and an inhibitory phosphate. At the same time, you have a phosphorylation and an activating phosphate. So here in the middle, what we have are the names of some of the kinases and phosphatases that are going to be important in this process. So we have the kinase WEE1, which is going to phosphorylate the kinase complex into the inhibitory site, and we have the CDC25 phosphatase that is going to be able to remove the phosphate added by WEE1, therefore activating the protein. So when you have the cyclin bound to the CDK and you have a phosphate present at the activating site, you will have an active molecule. But when you have a phosphorylation in the inhibitory phosphate, you will have an inactive protein. So this is how phosphorylation and dephosphorylation control the activity of the cyclin-dependent kinases. So let's take a look at this process in a little bit more detail. So what we have here, it's, for example, the mitotic CDK and cyclin M. They're going to bind together and associate. That complex is going to be inactive, except that you're going to have now the activation of the kinase WEE1. And kinase WEE1 is going to be able to phosphorylate the CDK in the inhibitory phosphate. To allow activation, you need to remove that inhibitory phosphate by having the activating phosphatase CDC25 remove the phosphate, letting you have the activated M-CDK. This example shows you here the actual timeline of the events. So these names are looking at the CDK and cyclin complex in yeast, Saccharomyces pombe. And we're going to be looking at the G2 to M checkpoint when we have the activation of the M-cyclin. Now, What we have over here are proteins called CDC. CDC stands for cell division control. Those are the name of the mutants that were initially isolated from S. pombe and Saccharomyces cerevisiae that control cell cycle. So the mitotic cyclin is called CDC13, and the cell cyclin-dependent kinases is called CD2. Now, you can appreciate from this image that CD2 contains two different sites for phosphate, a tyrosine at position 15 and a threonine at position 161. The first step in the activation of the mitotic kinase is going to be the binding of the cyclin to the cyclin-dependent kinases, and that is going to form what is called a mitosis promoting factor, or MPF. So just binding of the cyclin to the CDK complex molecule creates a complex, but that complex is inactive. Here is when we have now the phosphorylation of the molecule. So we're going to have one phosphorylation molecule, a kinase called V1, as I mentioned, and V1 is going to phosphorylate at position tyrosine 15. Another molecule, it's going to be CAC, and CAC is going to phosphorylate the protein at position threonine 161. This molecule has a phosphate at the inhibitory tyrosine position, tyrosine 15, and the stimulatory or activating threonine 161. And therefore, because the phosphate at position 15 is present, the molecule is inactive. Now, CDC25 is the phosphatase, and that's going to remove the phosphate added by V1 at tyrosine 15. Once that phosphate is removed, and you have the phosphate present at threonine 161, that leads you to an active mitosis promoting factor, which is then able to phosphorylate and activate other proteins. The active side of the molecule is composed by both a little bit of the cyclin and a little bit of the CDK molecule. So that is one way in which the activation of the cyclin-dependent kinase complex is activated by phosphorylation and dephosphorylation. So again, you have the initial binding number 1 of the CDK to the cyclin, and then the phosphorylation and dephosphorylation of the regions in the cyclin-dependent kinase. But that's not the only way in which the molecule can be inhibited. Once active, the cyclin-dependent kinase complex can be blocked by CDK inhibitors. In this case, you have an example of P27. P27 is a protein that will bind to an active CDK complex and therefore inhibit its function. So we're going to see that during the initiation of different stages of cell cycle, that proteins can bind to active cyclin-CDK complexes to inactivate them temporarily. And once the function has been performed, that protein can detach, allowing the molecule to go back to its active state. So let's take a look at this in a moment. So we're going to be looking, for example, at the cell cycle, and we're going to be looking at the transition from G2 into M. So we ask the question, is DNA replicated? Is all the DNA damage repaired? And if the cell can answer yes to both those questions, that cell will enter then the M stage and enter mitosis. So one of the things that it's required for the passing of that stage, it's as you can see, the destruction of the cyclin. And the destruction of the cyclin is mediated by ubiquination. So you're going to have the activation of an APC, which is an anasase promoting complex, and that APC complex is able to initiate and target the cyclin by ubiquitin. Once ubiquitin proteins are being attached to the cyclin, that cyclin is targeted to destruction, and therefore the amount of cyclin goes down, as shown here in the top. So that is the third way in which the cell reaches control of the cell cycle. So in this image, what you have is the S-phase CDK that is going to bind to the S-cyclin. that is going to eventually be phosphorylated and dephosphorylated, allowing to have an active S-CDK complex that is going to trigger DNA replication in the cell. And eventually, the S-cycling in that complex will be targeted by ubiquitin for degradation, releasing now the inactive S-phase CDK. Now, the mitotic CDK is present as well in the cell, but not the mitotic cycling. Once the mitotic cycling now is present, the M-cycling is able to bind to the mitotic CDK, producing eventually after phosphorylation, producing an active M-CDK that is now going to trigger the mitosis machinery to occur. Eventually, the M-cycling will be ubiquitinated and destroyed at the end of M phase, and the inactive CDK will remain in the cytoplasm ready for it to be used later. So this cycle of activation and inactivation of the cyclin and cyclin-dependent kinase complexes happens by phosphorylation as well as by the binding of the cyclins to the appropriate CDK, as well as the destruction by ubiquitination of the cyclin after the cell cycle stage has been finished. So let's take a look at some of those checkpoints. As I mentioned to you, the checkpoints are designed to question the cell to see if the events that are happening in each stage of the cell cycle has been accomplished, and if they are, they're going to allow that cell to proceed down to the next phase of the cell cycle. So what we can see is that different inhibitors are going to allow for the stop of the progression of the cell cycle. So for example, at the beginning, if a cell is going from the G1 to the S phase, you are going to have a CDK inhibitor that is going to block the G1 kinase to prevent it from going into S phase. You can also have inhibition of the CDC25 phosphatase that is going to then prevent the movement of the cell from G2 into M phase. And at the end, you're going to have the inhibition of the APC, or the anaphase promoting complex, that is going to prevent and delay the cell from exiting mitosis. Another component that the cell may have is a different pathway. So in a cell that is in G1 state, if it is going to be inhibited and unable to go into the S phase, for example, if the situation for that cell, the environmental situation is not appropriate and the cell doesn't have any nutrients, it can start the G0 state. The G0 state is a modified state in G1, and that is going to be a temporary resting state for the cell. So some cells are going to be able to go into the G0 state as part of the normal development and therefore not divide anymore. Other ones are going to be able to induce entry into the G0 states if the growth factors that they require are missing. But that is an alternative stage in which the cell can go instead of progressing to the cell cycle. So the G0 state is a quiescent, non-dividing cell state. Now, the APC molecule that I mentioned you here is the anaphase promoting complex, and that is going to prevent mitosis to occur until all the chromosomes are ready to be separated into each of the daughter cells. So different proteins are going to be related to different checkpoints to make sure that the cell either fixes its problems before it goes into the next cell cycle stage, or finds an alternative route like the G0 route until the conditions are better. So now let's take an example of how a cyclin and cyclin-dependent kinases control cell division. And in this one is going to be the sample of how S phase is controlled by the cyclin and the S-CDK. So as you know, DNA synthesis is occurring during S phase, and what you're going to have are two things. Number one, that the DNA is replicated without mutations, and that the DNA replication only happens once. The cell doesn't want to include its ploidy by having more DNA than it needs. So that is going to be controlled, as you know, by proteins binding to the origin of replication. In the chromosome, the origin of replication is the DNA area where the DNA will be, replication will be initiated. So now, that is bound by an origin of replication complex in the DNA. This image shows you here the process that is going to happen during the transition between G1 and S phase. So what we have here is a DNA molecule shown in orange, and bound to that DNA molecule it's the ORC, the origin recognition complex, and that is sitting on the origin of replication during all the phases of the cell cycle. So it is a complex that is attached to the cell DNA all the time. Now, as the cell begins to pass through the G1 state and the cell is getting ready to replicate its DNA during S phase, what you're going to have is binding of ORC by a protein called CDC6. So CDC6 is able to bind to ORC, and when it binds to ORC, it's able to help recruit DNA helicase into the origin of replication. Now that you have this image is actually not completely correct because it's showing you that CDC6 it's dissociated now that helicase is present, and in reality it's not. So pretend that this part here in the middle is not really happening, but it's going to be continually bound over here to the ORC. Now, what is happening here is that the cell is beginning to progress towards the cell cycle toward the S phase. So you're going to have the ORC, you're going to have helicase, and you're going to have CD6, and that forms what is called the pre-replicative complex, or pre-RC. That is the place in which the origin of replication now it's loaded with the helicase, which will help melt away the strands, but that complex cannot initiate replication yet. It doesn't have DNA polymerase. Now, when the cell enters the S phase, you're going to have an increase in the amount of S-cyclin. S-cyclin is going to be able to bind to S-CDK, and after activation by different kinases and phosphatases, you're going to have now the active S-CDK kinase. That kinase is going to be able now to do a couple of things. It's going to phosphorylate CD6, and by binding to CD6 and phosphorylate it, it's going to remove it from the complex. That it's going to phosphorylate ORC, and ORC that is phosphorylated can no longer bind to CD6, which then is removed as well. But it also is going to help recruit the DNA replication machinery, and that DNA replication machinery is going to then initiate DNA replication and having different replication forks, one here in the left and one here in the right. So that is how DNA replication is initiated. To avoid re-replicating DNA, since ORC is always bound to the origin, you need to have the ORC being phosphorylated by SCD6, SCDK, excuse me. Also, that is going to phosphorylate is going to phosphorylate CD6, which now phosphorylated, it will be targeted to degradation, and therefore it can no longer bind to ORC and recruit more helicase. At the end, what you have is that the phosphorylation of the ORC, it's going to prevent it from being bound by CDC6. You're going to have phosphorylation of helicase, which is going to activate it, but eventually is also going to export it out of the nucleus to prevent re-replication of the DNA. And as I mentioned, CDC6 is going to be also phosphorylated by SCDK to allow it to be now targeted to ubiquination and therefore degradation. So in that way, the activation of the cyclin-dependent kinase during S phase helps activate DNA replication, and only once. Okay, so in that case, what we have that at different times of the cell cycle, you're going to have different checkpoints. So for example, if the DNA intact, and if the DNA it is intact and non-damaged, the cell is able to progress down the cell cycle. But if there is DNA damage, in particular double-stranded DNA damage, you're going to have activation of different genes, including the protein P53. And you're going to also have the activation of the CDK inhibitory protein P21. Let's take a look at how that is happening to prevent the cell from entering into M phase. Now, as you know, DNA can be damaged in different ways, and one of the ways in the DNA can be damaged is by X-rays. That is going to induce a double-stranded DNA break, separating the two chromosomes. Now, I'm not going to tell you right now all the details, but what we're going to see is that you're going to have proteins that are going to recognize those ends of the DNA and be able to find it. And those little cascades are shown here by the multiple arrows. So what that is going to lead is to an activation of the protein p53. Under normal circumstances, p53, it's always degraded in proteasomes, but when it is phosphorylated, now the protein is stabilized and also activated. And when it is active, it can go inside the nucleus, and inside the nucleus it can bind to the promoter of the protein of the gene p21. The gene p21 is going to be then activated, transcribed, and translated into the p21 protein, which is a CDK inhibitor protein. So it's able then now to bind to the G1S or the S-CDK, preventing the molecule from being active. So by activating p21, you can now inhibit the progression of the cell cycle, allowing the cell to pause and have enough time to repair its DNA. So if the cell is able to repair the damage normally occurred here, the CP21 will be inactivated, and therefore the cell will continue down the cell cycle. But if the DNA is damaged beyond repair, now what you're going to have is the activation of pro-apoptotic genes that are going to lead to cell death and destruction of the cell. So let's take a look at apoptosis, since now we're looking at that point. So as you know, apoptosis is the process of programmed cell death, and it is a genetically active process in which the cell will have the DNA destroyed, and it will have the membrane be completely wobbled, and is required in different stages of development. For example, during embryonic development, it helps control the generation of certain structures, and in adults, for example, it allows for homeostasis, for the maintenance of cells at equilibrium. This is an example of how apoptosis is used in development. Here, for example, you have a frog that is juvenile, so it's going and developing. So from the tadpole stage, the frog now is beginning to expose his little feet that are beginning to grow. And between the digits of the frog, you have a membrane. That membrane forms a web, and that web is not really needed. So eventually that web, the cells that are connecting the digits in between that web are going to be activated by apoptosis to completely die, and therefore separating the independent digits as shown here on the right. Also, the tadpole has a long tail that it used to propel. So when the tadpole is now undergoing metamorphosis and maturation, that tail will be completely not needed anymore, and therefore the cells that comprise the tail and all the muscles that are used for locomotion will be eliminated by apoptosis. So that is a controlled way of death. That is different from necrosis, when a cell is destroyed, for example, by a virus or by physical shear stress, and now all the components of the cell are released outside of the cell. So, whereas apoptosis, everything is maintained within the cell, so the cell sort of shrivels into a racing. During necrosis, all the components of the cell are spilled outside, and that causes an alarm set in the organism because it's a stage of inflammation. So here we have a little image showing the stages of apoptosis or programmed cell death. So here we have a cell that is normal and is undergoing apoptosis. So you're going to have multiple things. You have to have a convolution. The cell is going to actually change shape. You're also going to have the compaction of the chromatin. So the chromosomes inside the nucleus are going to begin to be compacted and destroyed. Eventually, you also are going to have condensation of the cytoplasm, so the cytoplasm is going to begin to shrink. Following these steps, you're going to have the nucleus being fragmented and the blebbing of the membrane, as shown over here, by different portions, and also then cell fragmentation. Eventually, the cell fragments that are being generated can be phagocytosed by a phagocytic cell for elimination. So here in this photograph, what we have is a cell, a lymphocyte, normal, showing the different position of the membrane on the left, and on the right, a cell showing severe blebbing of the membrane, showing all this beginning of the fractions that are being formed as the membrane becomes to bleb. This image in this movie is going to show us some of the processes happening in vitro during apoptosis.
Apoptosis, a form of programmed cell death, has been induced in these cultured cells. Cell death is characterized by blebbing of the plasma membrane and fragmentation of the nuclei. Suddenly, cells weaken attachment to the substratum that they have been growing on and shrivel up without lysing. In the following movies, we observe the process at higher magnification.
So as you saw in that movie, the membrane of the cell begins to lose its shape. The cell loses its entire shape because the cytoskeleton that is maintaining cell shape has been destroyed, and therefore you begin to have cell blebbing. Now, normally, a cell may have a default pathway of death, and therefore it needs to receive signals that are going to allow it to survive, and those are called survival factors. Survival factors are going to activate Bcl-2, and Bcl-2 is a protein that is required to prevent cell death. For example, in the immune system, the cytokine IL-7, interleukin-7, it's going to induce survival of lymphocytes by activating Bcl-2 via the interleukin-7 receptor. So this is basically summarizing this image. A survival factor like interleukin-7 binding to its receptor, initiating a cascade of signal that is going to be transduced and therefore result in the production of Bcl-2 protein. And Bcl-2 will prevent apoptosis from happening. One way that we learned earlier about how to also prevent apoptosis is by the inactivation of the protein BAD. Bcl-2, which is bound by BAD, which is a Bcl-2 antagonist of cell death. So BAD protein binds to Bcl-2, maintaining it inactive. If BAD binds Bcl-2, the cell will die by apoptosis. So if you have the activation of the PI3 kinase system, that is going to activate AKT. And as you know, AKT, once activated, it's going to be able to phosphorylate BAD. Phosphorylated BAD is inactive. That releases then Bcl-2. And Bcl-2 will then be able to inhibit apoptosis. So the survival of a cell can be induced via the production of Bcl-2 as well as via the inactivation of BAD, which is preventing Bcl-2 from working. Now, if the cell is going to undergo apoptosis, you're going to have the set of events that are going to mediate apoptotic cell death. So let's assume that a cell is undergoing apoptosis, and you have the stimulus shown over here in the bottom left to undergo the apoptotic cell death. That is going to induce cytochrome C, which is usually present in the intermembrane space. As you know, it's part of the electron transport chain, to now be exported into the cytoplasm. That is going to be done by proteins called BACs or BAC, and BACs and BAC are Bcl-2 family members. Now, the release of cytochrome C into the cytoplasm from the mitochondria, it is a test signal. And what you're going to have, it's now cytochrome C binding to a protein called APAF1, or apoptotic protein activating factor 1. APAF1 bound to cytochrome C is going to assemble into a complex that now is able to recruit a caspase. And in this case, caspases are proteins that induce apoptosis, and they are maintained in an inactive state, maintained in a pro-caspase state. So in that case, what we're going to have is the APAF cytochrome C complex forming this subunit, and that is going to recruit procaspase 9. Procaspase 9 is going to be recruited and eventually activated, forming what is called the apoptosome. The apoptosome, now it's able to release caspase activation that is going to lead to apoptosis. So here we have the inactive caspases on the left, and the inactive caspases on the left are going to associate to one another, removing what is called these prodomains to allow the cleavage and assembly of the active caspases. So here in the left, we have two different procaspases. They're going to bind to one another. They're going to have two different cleaving, and those two different cleavings are going to remove the prodomain and allow the protein to rearrange in an active form. Now, those caspases are going to activate a cascade of apoptotic molecules. So one molecule that is an active caspase is going to be able then to mediate the proteolytic cleavage of other caspases, and those other caspases are going to now target some proteins, for example the nuclear lamins that are going to now destroy the nucleus, but they're also able to activate other caspases. So you have an amplification event. This amplifying cascade of caspases is going to now lead to the cleavage of many of the cytosolic proteins, including, for example, the actin filaments. And the actin filaments, when they're destroyed, they're going to allow the cell to begin the blebing process. This movie from the Weehai Educational Center in Australia shows the process of apoptosis. What we have over here is a tissue, shown here in pink. And this brown cell here at the bottom right, it is a disease cell. And what's going to happen to that disease cell is that a cytotoxic T cell, a killer T cell, is going to come around and give it a apoptotic signal by triggering the trimerization of a fast death receptor. So let me show you that, and I'm going to try to narrate a little bit since the video doesn't have narration. So again, normal tissue with a cell that is diseased. Here's the healthy cells, and here's the disease cell. Now we have a killer T cell that is scavenging around looking for cells which are not normal anymore. And here's interacting with the disease cell. The T cell has a death ligand on its membrane. Here you see the plasma membrane of the killer T cell approaching. And you see these green proteins, those are death receptors. So the death ligand is pink, and the death receptor are green, and you need to bind 3 death receptors. That has brought now the domains in the cytoplasm of those 3 receptors. We're able to bind adapting proteins, and those adaptor proteins are going to recruit procaspase 8. Here we have them activating procaspase 8 into caspase 8. Procaspase 8 is going to initiate the cascade by activating procaspase 3. Notice the amplification. Caspase 3 is going to go around now, destroying other proteins, initiating the apoptotic process. Here we have the mitochondria of the cell, and the apoptotic signal cascade is moving towards it. And inside the cytochrome C present in the mitochondria is released. Now, APF1 is able to bind to cytochrome C, and those APF1 cytochrome C complexes will bind to one another. And they're going to now recruit procaspase 9. That will create the apoptosis. Now, caspase 9 activated will initiate the activation of caspase 3. One of the targets of caspase 3 is the actin filaments that form the cytoskeleton. So as you see, the actin filaments are beginning to be destroyed, and that is going to begin to cause the cell to bleb. The cell no longer attached to the substratum, and the membrane becomes to bleb, generating the little particles that could then be phagocytosed by scavenger cells. So if you want to see the animation again, you can have, here's the website. I'm going to also load that movie to crops, so after doing that. And I believe that concludes this lecture. So if you have any questions, we're going to have a discussion about this on Monday. I hope that you had an awesome Thanksgiving, and I will see you very soon.